Cytotoxic T cells can induce target cells to undergo programmed cell death. Cells can die in either of two ways: physical or chemical injury, such as the deprivation of oxygen that occurs in heart muscle during a heart attack, or membrane damage with antibody and complement leads to cell disintegration or necrosis. The dead or necrotic tissue is taken up and degraded by phagocytic cells, which eventually clear the damaged tissue and heal the wound. The other form of cell death is known as programmed cell death or apoptosis. Apoptosis is a normal cellular response that is crucial in the tissue remodeling that occurs during development and metamorphosis in all multicellular animals. Most thymocytes die in apoptotic death when they fail positive selection or, much less often, are negatively selected as a result of recognizing self-antigens. Early changes seen in apoptosis are nuclear blebbing, alteration in cell morphology, and, eventually, fragmentation of the DNA. The cell then destroys itself from within, shrinking by shredding membrane-bound vesicles and degrading itself until little is left. A hallmark of this type of cell death is the fragmentation of nuclear DNA into 200 base pair BP pieces through the activation of endogenous nucleases that cleave the DNA between nucleosomes, each of which contains about 200 BP of DNA. Cytotoxic T cells kill their targets by programming them to undergo apoptosis. When cytotoxic T cells are mixed with target cells and rapidly brought into contact with centrifugation, they can program antigen-specific target cells to die within 5 minutes, although death may take hours to become fully evident. The short period required by cytotoxic T cells to program their targets to die reflects the release of preformed effector molecules which activate an endogenous apoptotic pathway within the target cell. As well as killing the host cell, the apoptotic mechanism may also act directly on cytosolic pathogens. For example, the nucleases that are activated in apoptosis to destroy cellular DNA can also degrade viral DNA. This prevents the assembly of virions and thus the release of infectious virus, which could otherwise infect nearby cells. Other enzymes activated in the course of apoptosis may destroy non-viral cytosolic pathogens. Apoptosis is therefore preferable to necrosis as a means of killing infected cells. In necrosis, intact pathogens are released from the dead cell, and these can continue to infect healthy cells, or can parasitize the macrophages that ingest them. And that cytotoxic T cells can induce target cells to undergo programmed cell death. Have a great day.